Meiosis I. In this module you will learn about meiosis I. Meiosis is a nuclear division that occurs in the germ cells of sexually reproducing organisms during the production of reproductive cells called gametes. Meiosis is a reductional division that reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by two cell divisions, meiosis I and meiosis II. The entire process of meiosis results in the formation of 4 haploid daughter cells with half the chromosome number as that present in the diploid parent cell. However, after fertilization occurs, the chromosome number gets restored. Meiosis I is the reductional division in which the chromosome number gets halved and distributed between two daughter cells. Meiosis I includes 4 stages: prophase I, metaphase I, anaphase I, and telophase I. Let's know about the homologs before studying the phases of prophase I. Each diploid cell has 2 copies of chromosome, one inherited from the male parent and the other from the female parent. These 2 copies, coming from different parents, are similar to each other and are called homologs. Now let us look at the different stages of prophase I in detail. To begin with, let us learn about leptotene. During leptotene, the chromosomes condense and become visible. The zygotene phase is followed by pachytene. During pachytene, the chromosomal crossing over now takes place between the non-sister chromatids of the homologous chromosomes. This crossing over is what makes each individual unique and causes the recombination modules to be visible. As the crossing over completes, it marks the end of the pachytene phase. Diplotene is followed by diakinesis, which is also the last stage of prophase I. During diakinesis, the chromosomes are fully condensed, the nucleolus and the nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase I is followed by metaphase I. During metaphase I, the microtubules from the opposite poles attach to the chromosomes at the kinetochores, and the bivalents align along the equatorial plate with a random orientation. Either of the parental homolog takes up any site on the spindle. The metaphase I is followed by anaphase I. During anaphase I, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules. The sister chromatids, on the contrary, remain attached at the centromere. In this module, you have learned meiosis is a reductional division which reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by 2 cell divisions, meiosis I and meiosis II. Meiosis I includes 4 stages: prophase I, metaphase I, anaphase I, and telophase I. Prophase I is further subdivided into 5 stages: leptotene, zygotene, pachytene, diplotene, and diakinesis. During leptotene, the chromosomes condense and become visible. During zygotene, the duplicated chromosomes pair with homologs to form bivalents. During pachytene, the attachment of homologs to each other along their length, that is synapsis, is complete. In diplotene, synapsis brings to breakdown, the chromatids start to pull apart, and chiasmata are visible. By diakinesis, the chromosomes are fully condensed, the nucleolus and nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase I is followed by metaphase I, when the microtubules from opposite poles attached to the chromosomes at the kinetochores, and the bivalents align along the equatorial plate. During anaphase 1, the homologous chromosomes separate out and move to opposite poles with shortening of the microtubules, forming two haploid sets. During telophase 1, the chromosomes reach the poles and nuclear envelopes are reformed around the two daughter nuclei and the nucleoli reappear. A furrow appears in the cytoplasm and the cell divides into two by cytokinesis.